All right, I'm back and it's time to build some DNA. So it's very exciting. This is going to be the next part of our DNA replication discussion. And so we're going to go into the details about how we're going to replicate a piece of DNA, and then we'll talk a little bit about how mutations can occur. So we've already talked about DNA replication. When is DNA replicated? Of course, that's during S phase of interphase, right? Synthesis phase. So that's when these steps occur. Why is it replicated? Remember, if you're making new cells during mitosis, each cell needs a full copy of all the DNA. Or if we're cells are making gametes through meiosis, each cell needs half a copy of the DNA, but we're going to make 4 cells. At any rate, we're making new cells, and every cell has all the DNA that you contain, that the organism has. You know, again, except, you know, obviously the gametes have half the DNA, but, you know, every cell has all the DNA. So again, this is you know, I always think that's just an amazing fact, right? In human cells, again, it's like 3 meters long total DNA, and you've got, you know, 1 trillion cells, and every one of them has got 3 meters of DNA. It's an incredibly thin molecule. But, you know, again, this means like, you know, your pointy finger, right? It doesn't have just pointy finger DNA. It's got all the DNA in it. Your eye cells, your brain cells, you know, they don't have just brain cell DNA. They've got all the DNA. At any rate, we've got to make copies of the DNA so that each cell can have a full copy. You'll also remember that if I know one strand of the DNA, then I automatically know the other strand, because all tigers can growl. A is with a T and C is with G. And so it's this fact that allows me to make a copy of any DNA molecule. I only need 1/2 of it, and I can make the other half. So again, you'll remember that during interphase, each chromosome is stretched out and is incredibly thin. You can't see them with a microscope. And after S phase, each one of these stretched out chromosomes has been replicated and has made an exact copy of itself. And so now you have 2 sister chromatids, and they're joined at a centromere. And so in this example, here we have a very, very, very small piece of DNA. And we can separate that DNA. Remember, it's hydrogen bonds that's holding together these 2 strands. And although there's a lot of them, they're still weak. And so it's pretty easy to break them and to pull these strands apart. And so we can do that. We can separate the strands. So if I separate those strands, I can look at each one individually. If I have an individual strand of DNA, I can, of course, make the complementary strand. So again, in this example, from top to bottom, what would be the complementary strand? The first nucleotide in our template is A. So our first nucleotide from top to bottom in our complementary strand must be a T, right? Well, look at all the choices. Only one of them starts with a T, so we can eliminate everything. It has to be choice B, right? And so again, if you look at each of those nucleotides, they match up properly, going top to bottom. And I started with a double-stranded DNA, and I separated the strands. Well, now I can do this. I can do the A with T, C with G for each of those strands. And so for each of those strands, I can fill in the complementary strand for each of those original strands. That's DNA replication. So if you look at the start here in A, we had 1 DNA molecule, and then at C, now we've got 2 identical DNA molecules. And so it's that simple. That's how we're able to make copies of all our DNA. And every time you make a cell, your cells do this. and get it right every time. That's pretty awesome. Now look at the coloring, what they're trying to emphasize in this figure. We started off, and the original nucleotides and the original strand are this dark blue. And so then in section B here, we've separated them, but it's still dark blue. But then the new strands that we built are this kind of light blue. Those are new nucleotides. So the dark blue is old nucleotides, original nucleotides. The light blue are new nucleotides. And so in each of those new strands, it's half the original strand and half new nucleotides. And so we call that semi-conservative. So this is just one of those things that's interesting and helped us figure out how DNA gets replicated. So when you replicate DNA, you could end up with two strands, one of which is the original strand and one of which is all nucleotides, but that's not how it works. You start off with the original DNA, rip it apart, the two new ones are half the original DNA and half new nucleotides. Semi-conservative. Okay, so how does this all begin? You've got this long piece of DNA, and you have origins of replication. You got to start somewhere, and that somewhere we call origins of replication. And so the first thing you got to do is you got to start to separate those strands, right? So at this origin of replication, we pull apart the DNA strands, and we make a replication bubble. And so that's what they're trying to show you here. So on the left, it's showing you the original strand, and then several of these bubbles, and the bubbles grow until you've got two new strands. And again, on the left, if you notice, the original strands are dark blue, the new nucleotides are light blue, and so at the very bottom, we've got two identical pieces of DNA, and they each have a dark strand and a light strand, because they each have one half is original DNA and one half is new nucleotides. Now what the point we're trying to make here about these replication bubbles is, you don't just start at one end of DNA and then zip through all the way to the other end. That's not how it works. You start in several places on the DNA and you go in both directions. So if you look at the pink arrows, that's showing the direction that you're going. So you start with these small bubbles, and the bubbles grow. The bubbles get bigger. And then you have many of these bubbles that grow, and then eventually the bubbles meet. And when all the bubbles meet, now you've got two new identical pieces of DNA. And so the picture on the right is just a microscope picture kind of showing that, and you can kind of see where you've got different bubbles. But this is important to understand how the DNA gets built. Is you start at that origin spot and you go in both directions. And then you start in other places and you go both directions, and eventually all those bubbles meet up. Okay, so you start by unwinding the DNA. Remember, it's a kind of a twisted ladder. And so you've got an enzyme called DNA helicase that unwinds the DNA. And so there are several different enzymes that take place. Excuse me. And when you get into genetics, you'll learn there's a lot more. You learn a lot of details about these. But the enzymes that I mentioned here are the ones that we want you to know. The most, we're going to talk about the big players here. And so what we're doing is we're going to look at one of these replication forks where the bubble is growing. We're going to zoom in on one of those. And so here we've zoomed in on one of those, okay? And so they're just showing you this drawing of helicase in green, and it, unzips the zipper. It unwinds. This is what kind of unwinds and pulls the strands apart, basically, is its job. Opens up the strand is another term we use. Something that we don't mention, I don't mention here so much, but you see there's single strand binding proteins. Once the DNA gets pulled apart, these other proteins come on and attach so that it won't come back together. Because you can pull it apart, but then it'll just want to come right back together. and we don't want that to happen. And so those single strand binding proteins help hold it open. And so once you have the DNA split open, the first thing that comes in is a piece of RNA. That's kind of cool. But a little piece of RNA, which is called a primer, comes in and attaches to a particular spot, the starting point on the DNA. And that's just a signal that, hey, this is where we're going to start replication. And so the big player in here is an enzyme called DNA polymerase. So DNA polymerase is a very important enzyme. It reads the DNA base by base and brings in the proper nucleotide and then sticks that nucleotide into the growing strand. And so the DNA polymerase will read one strand and says, oh, this is a C. I need to bring in a G. And so it will grab a G nucleotide and bring it into the new strand. And then it'll move over and read the next nucleotide. Oh, this is an A. I need to bring in a T, and so on. So DNA polymerase is a big player here. And so in this picture, they're showing the RNA primer that's first attached, and that tells DNA polymerase where to start. That's added by an enzyme called primase. And so here's just we zoom back a little bit now, and here you can see one of those growing replication bubbles. And in this example, the little red pieces are those little RNA primers that tell DNA polymerase where to start. And then the light blue arrows are the new nucleotides. So that's where DNA polymerase has brought in new nucleotides, but it doesn't bring them in at random. It reads the dark blue nucleotides, and it knows what complementary nucleotides to bring in. And so that's what they're showing you here. They're showing you DNA, and so it says DNA Pol 3. And so when you get into genetics, you'll learn there's lots of polymerases and different organisms do different things. But this example, DNA polymerase 3, is bringing in those new nucleotides, and you can see again in this example they're light blue colored, and it's attaching them to the dark blue ones, and in the process it's building a new strand, and it can do that on each strand. So it's reading each strand and bringing in the proper nucleotide. And so here we're back again. Let's look at the details a little bit. And yeah. And so here is where that directionality comes in. We made a big deal about each strand has a 5' end and a 3' end. When you're building a new strand, you always build it from 5' to 3'. And so look at the very last nucleotide in this example. At the very bottom, you see that nucleotide has got that 3' carbon, and it's just sitting there. There's nothing attached to it. And so if you look on the right where we've zoomed in on a nucleotide, you can see the nitrogenous base on the 1' carbon, and the phosphate is already attached to the 5' carbon. So the 1' and the 5' are already taken up. They can't, nothing can, there's already something bound there. But look at that 3' carbon. It's open. There's nothing attached there. And the only place you can find, and so there's nothing attached there, so that means something can come in and attach there. That's where the new nucleotides are going to come in. Well, the only place you can find such an open carbon is at the 3' end of the strand. If you look at this strand here, the 5' end, well, there's a phosphate on there. There's always a phosphate on the 5' end. And so the new nucleotide is going to come in and attach to that 3' carbon. And so we can only build a new DNA strand by starting at the 5' end and elongating toward the 3' end. Now again, we're not just building this at random. We're bringing in these nucleotides by reading the other strand. We're only looking at one strand here. But every new nucleotide must be brought in and attached to that 3' carbon, to that 3' end. And so the new nucleotide, it already has a phosphate on it. That phosphate is very reactive. It's going to come in and attach to the 3' carbon of the growing strand. So the growing strand has an open 3' carbon at the very end. This new nucleotide comes in. We're going to add that new nucleotide. DNA polymerase is going to say, oh, hey, I need a cytosine, a guanine, whatever. It's going to bring it in, and the phosphate from that new nucleotide is going to attach to that 3' carbon, and now the new nucleotide has an open 3' carbon. So we're always adding to the 3' end. Now, just a little digression here about this reaction, about how those nucleotides are brought in. Look at this nucleotide. This is an adenine nucleotide, which is ready to be incorporated into the new growing strand. So DNA polymerase, reads, oh, thymine, I need an adenine. Oh, here's an adenine right here. And I'm going to take this adenine and I'm going to stick it to the new growing strand. But look at what we have here. We don't have one phosphate, we actually have 3 phosphates. Well, does this look familiar? Do you remember this? This is an adenine with 3 phosphates. It's adenosine triphosphate. This is ATP. You've already seen this, right? You remember that ATP is a nucleotide, and those 3 phosphates have a lot of energy in them. Well, we're going to use that energy. It's very reactive. That's what we're going to use to help catalyze this reaction. And we're going to bring those 3 phosphates in, lose a couple of them, and that phosphate that remains is going to attach to the growing strand. And we can actually do this with guanine, cytosine, and thymine too. They can also have 3 phosphates on the end and be reactive. And so that helps to catalyze this reaction. So that's just a little side note. And this is that figure we looked at before, that those 3 phosphates have a ton of energy that can drive reactions. Well, this is an example. We're going to use that energy to help build a new DNA strand. And we're going to use the ATP itself as one of the building blocks. But again, those are attached to the 5' carbon. And so they're already on the 5' carbon of the new nucleotide. They're going to react and attach to the open 3' carbon of the strand that's being built. And so I made a big deal about this, but again, this is one of those things you hear all the time when you talk about DNA replication. It grows from 5' to 3'. Well, this is why. This is why it always grows in that direction. Because you always got to bring in the new nucleotide to that open 3' carbon. And so I made a big deal out of this. Just, it's one of those things you got to remember. Like, does it grow from 5' to 3' or 3' to 5'? I can't remember. Well, if you can kind of remember this structure of the nucleotide, you can work it out. You can say, oh yeah, if I remember that what a nucleotide looks like, that 3' sugar is always open. And that's where new stuff has to come in. And so we always have to grow toward the 3' end. Another completely different way to remember this is 5, 4, 3, 2, 1, synthesize, right? And so you go from 5 to 3 to synthesize. Whatever works for you, but you always grow from 5 to 3. And so that's kind of what they're showing you here. This is a figure from your book, and if you look, all the strands that have arrows on them, those are all growing strands. Those are being synthesized, and if you look at how they're labeled, all those growing strands are growing from 5' to 3'. And that's what I just said. Now, consequently, the template strand, the original strand, the strand you're reading, it must always be read from 3' to 5', right? So we're moving along the template strand from 3' to 5', but we're building the new strand from 5' to 3'. So, try and keep that straight in your head. Now, you'll notice something else about this picture. here we've opened up that strand, that replication bubble, and all new strands must be built from 5' to 3'. And so if you look at the bottom one, what's labeled, it's called the leading strand. Look, you see it's just a big smooth strand. 5' to 3'. Let's just keep building. 5' to 3'. Let's just keep building it, right? But look at the other one, what's labeled the lagging strand. It's not very smooth. It's built in these little short chunks. And so you build a chunk, and you open up the bubble, and you build a chunk, and you open up a bubble. And you have to do it. It has to occur that way because you have to build these from 5' to 3'. So one can be easily built from 5' to 3'. The other can only build a little at a time. And so as you open up the bubble, right, as that bubble gets bigger, you can see that replication fork, that's where we're separating the strands. That leading strand, you can just keep trucking. As you open it up and unzip it, that leading strand just keeps going because it's going 5' to 3'. There's no reason to stop. But in that lagging strand, you have to build in the other direction. And so you have to open it up, and now you can build a little bit, and then you have to open it up, and now you build a little bit, and then you open it up and you build a little bit. So it's built in fragments, and that's called the lagging strand. And so in this picture, they're showing you that same thing in just a different way. That you can see how you've got some of these strands are just one smooth piece. They're easily built in the 5' to 3', whereas the other strands are built in little chunks. And so this would be a great quiz question, for example. I could give you this picture. You see that none of these strands are labeled with their 5' to 3', but you should be able to look at this, and for every one of these strands, the dark blue and the light blue strands, you should be able to label the 3' and the 5' end based upon what you see here. So if you can do that, then you kind of understand what I'm getting at. And again, why am I making such a big deal about this? Because it is a big deal. Like understanding how this DNA gets built, that's really important, and it really helps us understand a lot about DNA. So it's these kind of details that we unlocked in the 20th century, and now we routinely use in the 21st century to understand more and more about biology. Now remember, we opened up the original strand at those origins of replication. And then we unzip the DNA in both directions. And so you see the overall directions of replication, we can go both ways. And so we open it up and we start to copy each strand from that same spot, but we can go in both directions. So it's a lot faster. If we had just started at one end and went in one direction, it would take way too long to copy the DNA. And so again, leading versus lagging. The DNA polymerase can start at the point marked there, and it can just keep going as the bubble's unzipped. But in the other direction, you have to unzip a little, build, unzip a little, build, unzip a little, build. And the same thing happened on the other strand, but in the opposite direction. Okay. Well, I've beaten that idea to death, but that's an important idea. At that point, after all that, if that continues until all the bubbles meet, then guess what? You've got two identical strands of DNA. You've just replicated your DNA. You've just fulfilled what you're supposed to do in S phase. And so now I've got all my chromosomes replicated. Now I can go through mitosis or meiosis and divide those chromosomes in a proper manner. That's it. That's it. All right. Last thing we need to talk about is mutation. What happens when something goes wrong? You remember that a mutation is when the DNA changes. So one of the things that occurs as DNA polymerase is building these new strands of DNA is that they proofread. The enzyme can tell, and it kind of goes along and it makes sure that the right nucleotides are in the right spot. But every once in a while, it makes a mistake. I mean, these are just chemical reactions. I say it proofreads, or it goes and grabs, like we sort of personify these molecules like they have conscious thought. Of course they don't, right? These are chemical reactions. And sometimes things can go wrong. And so when this happens, you can get a change in the DNA, and that's going to ultimately change the RNA, and that might change the protein. Well, that change in the DNA is a mutation. And so what can mutate the DNA? You can be exposed to harmful chemicals. There are a lot of chemicals that make it more likely for the DNA to be changed. Cigarette smoke or high energy X-rays. All those things can cause the DNA to change, but that also can just happen. Sometimes that's life. Sometimes the reactions are just imperfect and it just happens. And so in this example, we're looking at repairing the DNA as we synthesize it. And so at the very start, there's an incorrect nucleotide. Now, think about what I said earlier. You always have a single ring nitrogenous base pair with a double ring, right? No matter what. A with T or C with G. It's always a double ring and a single ring. And so if there's a mistake and you get, say, like 2 double rings together, what's that going to do to the DNA? It's going to cause it to bulge out. Or if you have 2 single rings, they're going to bulge in. That is the sort of thing that an enzyme like DNA polymerase can notice, that there's something not right. Now, what caused that? Maybe it was an X-ray, maybe it was just a spontaneous, maybe it was a chemical. Something caused the wrong nucleotide to be stuck in there. And most of the time, DNA polymerase can recognize, hey, these are not matching up properly. It can go and cut out the bad part, stick in the right nucleotide, and no mutation occurs. No harm, no foul. But every once in a while, it doesn't get fixed. The wrong nucleotide gets stuck in. And so now we've mutated the DNA, and now we have a new allele. Remember what we said alleles were? Different forms of the same gene. And so if you have a mutation where you change the DNA, now you've got a different form of that same gene. You've got a new allele. And this is how we get new alleles. Okay, let me give you an example here. Pretend this is a single strand of DNA, and we want to make the complementary strand. So again, A's with T's, C's with G's. And so if I have one strand, I can easily build the second strand. We just talked about that. This is how we make exact copies of all our chromosomes. This is what DNA polymerase does. Well, pretend that DNA polymerase makes a mistake. And so it stuck the wrong nucleotide in. So if you look here, this T, this thymine that's in the blue box, that's not the right nucleotide, right? The polymerase read a G and it stuck a T in there. That's not going to work. That needs to be fixed. And usually it gets fixed properly. Usually polymerase recognizes that, takes out that T, which was wrong, puts in a C, which is right. No harm, no foul, no mutation. But what if, when DNA polymerase corrects this mistake, it fixes the wrong base? So in this example, it's supposed to cut out that T in the blue box and replace it with a C, but instead, it thinks the T is the right one, and it cuts out the original nucleotide and puts in an A, right? And so it's fixing its error, but it fixed the wrong nucleotide. And so that's another way, that's a way that we can then change the underlying DNA and get a mutation. And so on the left, you've got the original chromosome. On the right is the new chromosome, where we had stuck in the wrong nucleotide and then fixed the wrong nucleotide. And so that's how you can get a mutation pretty easily. But these mutations are very important in biology. So what happens? We just mutated the DNA. What can happen? You know that then the resulting RNA is going to change, because the DNA builds the RNA. And if you change the DNA, the RNA will change. And that RNA is going to go make a protein. Now, maybe that change in the RNA will change the protein. Maybe that protein is a little bit better. Now, what do I mean better? Okay, that's a relative term, but by better I mean better for the organism. Enhances its survival. And so most of the time these proteins are enzymes, right? Well, maybe the enzyme works a little faster. Maybe the enzyme holds its structure a little bit better in high temperatures. Any one of a million different ways that maybe that protein is just a little bit better. Maybe the RNA changes, but the protein stays the same. Even though the RNA changed, the protein might not change. And when we talk about protein synthesis in the next lecture, you'll see what I'm talking about. But it's possible to change the RNA and the protein doesn't change at all. So, although you mutated the DNA, nothing changed. Maybe the protein gets worse. And proteins have a very specific shape and a very specific primary structure. Their amino acids have to be in a certain order. And a lot of times you change up that order. You swap out an amino acid, that really messes up the protein. So you remember earlier we gave an example of sickle cell anemia, where you change one amino acid in one subunit of the hemoglobin protein, and that hemoglobin really radically changes shape. So that's one thing that you have to realize when we talk about mutation. In general, that word mutation has a very negative connotation, and it should. A lot of times these mutations are deleterious. They're bad for the organism. When we talk about the zombie apocalypse, oh, there was a mutation and it caused these whatever. But it doesn't have to be so. Mutations can do nothing, and every once in a while mutations can actually make a new trait that's better for the organism. Anyway, the bottom line is, when you make new cells, every one of the new cells has to have a full complement of the DNA, and so we've got to make a complete copy of all the DNA, and that was why we talked about DNA replication. To do that, we split apart an existing chromosome, and each individual strand acts as a template for the new second strand. But sometimes mistakes are made, and those mistakes are mutations, which may or may not affect the organism. Now, on the evolutionary time scale, these mutations are incredibly important, because this is how we get new traits. And so all the different alleles that you know of, and we can talk about them forever, but if the simple ones, if you talk about eye color, you've got a blue allele and a brown allele, maybe a green allele, right? Where did they come from? There was an original eye color, and at some point there was a mutation which caused a different allele. Well, this occurs for all genes, not just eye color, but all kinds of stuff. All the DNA can mutate. And every once in a while, that mutation causes a new protein that maybe works a little better, or works a little differently, or does something. But this is how we get new traits. And it's those new traits that natural selection can act upon. And so, if the organism that has the mutation gets a new character, and if that new character gives that organism a slight advantage, gives them better fitness, then they're more likely to survive, because they're just a little bit better at something. And if they're more likely to survive, then they're more likely to reproduce. And if they're more likely to reproduce, they're going to pass that new character on to their offspring, and their offspring are going to have this new character, and their offspring are going to have this new advantage. And over the millions and billions of years of evolution, this is how we get new traits and how new organisms evolve. And so, of course, we'll talk about that in a lot more detail when we get into evolution and natural selection. But that's why mutations are important. So, there it is. We've gotten to a little bit more detail about DNA and its structure and how it's copied, and we're just going to keep building upon this knowledge as we work through this chapter. But this is all, this is what makes biology so exciting, in this century. You know, they say that 21st century is going to be the century of biology because, you know, no science is going to grow as quickly as biology. Well, we can argue about that. Who knows? But this is why we are, you know, our knowledge in biology is growing so fast right now is because we're starting to understand the DNA molecule, and that's very exciting. And I just think back, you know, back to when I was where you are, when I was an undergrad, sitting in that seat, and what we've learned just since then, just in my academic career. It's amazing. So it's very exciting. You know, it's very interesting. And so we're going to, you know, learn more details about this. But anyway, I hope that you can get as excited about it as I do. If you've got any questions, of course, always let me know. And I'll see you later.